All right, continuing on. And I'm just going to short circuit a few steps here. Oh, Miss Ilio, what are you up to? We're still in the chordata, but now we're in the subphylum Urochordata. And Urochordata means the notochord. Remember, cephalochordata meant the notochord was in the head. In the Urochordata, that means notochord in the tail. And in the cephalochordata, the notochord did extend into the tail, but it also extended into the head. But the opposite is not true here in the Urochordata. In the Urochordata, we're going to find the notochord is only found in the tail, the posterior region, okay? And these animals are typically what we find are tunicates, are urochordates. Okay. This time, what we find in the Urochordata is they don't retain these four synapomorphies of chordata throughout their lives. They typically just retain them when they're larvae. The larvae are very small, but I'm going to draw it kind of biggishly. So I can emphasize things. Okay, so this time, this is going to be our anterior direction. This is going to be our posterior direction. This is going to be dorsal. And this is going to be ventral. All right, so when I drew our lancelet, it was swimming the other way. Our larval tunicate is going to be going in this direction. Whoop. Okay. So most of the business of staying alive is found in this anterior region. And I'm going to draw kind of like there's a chamber here. And they're kind of plumbed in a not exactly the same way as a lancelet. But hopefully you can see, based on how I'm drawing this, this is something similar to what we saw in our lancelet. Okay, so this whole structure here, this is our atrium. This back here is our stomach. And this thing here is our pharynx with gill slits. And sometimes it's referred to as a pharyngeal basket. And we'll see that in the adult form of the tunicate. Okay, so food comes in here, but we don't really think of it as a mouth so much as we call it an in-current siphon. So in-current, water goes in, just like we saw in sponges with the ostia. And this back here is our X-current siphon. So again, we're filter feeding. The food comes in through the in-current siphon. Water is going to keep flowing through, but any food particles are going to get caught in that pharynx with the gill slits, passed along into the stomach where they're going to be digested, and then waste is going to be passed out with flowing water through the X-current siphon. Okay, so now in the most dorsal direction, I'm going to draw this is going to be our D, T, and C. What does that stand for? Dorsal tubular nerve cord. And then kind of along the midline, we're going to have our cartilaginous rod that's only going to be found in the posterior end. And that's going to be our stiffening notochord. Okay, so that's our cartilaginous rod found only in the tail. Again, we're in the Urochordata. So this is the larva, which retains all four of these characteristics. I'm also going to draw an adult tunicate, which changes quite a bit, which is something we expect when we have a larval organism and an adult. So what we find with our adult tunicate is you might have seen these when you're out, if you're out snorkeling or scuba diving, and you could be forgiven if you were like, oh, it's a sponge. But unlike a sponge, it only has two openings. It retains the in-current siphon and the ex-current siphon. So this is going to be our in-current, that's supposed to be an arrow. In, one N, current, two R's, siphon. And this is going to be our ex-current siphon. As adults, these types of tunicates are sessile. They don't move anymore. These are the larvae are motile. As adults, they are sessile. There are some motile tunicates as adults, things like salps, but that's not what we're drawing right now. Okay, but what we do want to have in here is a fairly large gill slits a little bit bigger, a little bit more prominent inside of our atrium. So this opening here is still our atrium. And we still have this time I'm going to call it a pharyngeal basket with gill slits. So similarly, it's going to be filtering out food particles. And as an adult, it's going to loop down. So we've got our gut, getting a little bit more sophisticated here. So our pharyngeal basket is going to filter out food particles. It's going to send them down to a stomach. This body cavity is now a coelom, because these are sorry, not vertebrates, they're chordates, just like you and me. And we are eucoelomate, so there's going to be this body cavity, this coelom, that's separate from the atrium, and that's going to lead to an anus. So there's no postanal tail as an adult. There's also no notochord and no dorsal tubular nerve cord as an adult, which is what this is down here, an adult tunicate. So these animals These adult tunicates, you can find them typically in marine habitats. They usually have just these two openings, an in-current siphon, an ex-current siphon. You could be forgiven for thinking they were sponges. The tunic is an outer covering of cellulose. That's called the tunic. Tunics on a tunicate. But again, these are not animals that you would just expect to find just walking down the street. Oh, look, it's a tunicate. But they are important because they are part of our phylum, and they tell us a lot about how development can change from having a larval stage that has all four characteristics, all four chordate synapomorphies, to having an adult stage where that's not the case. And that's pretty much all I want to show you about tunicates.